ये कहिबे सब सेरा कांदे के पलाई रेडी ना के पलाई नेक्स्ट के पलाई मिलुराम सर
Good afternoon, all dignitaries. This is our last session of International Conference on New Horizons on Pharmaceutical Research 2021, organized by KANAK Institute of Pharmaceutical Sciences, ROURKELA, in association with ISTE New Delhi, India. This is our during this session we have kept the talk of our one keynote speaker. Along with we have our last valedictory session. Before that, we have some participants are there. They have to present and they have to contribute and share their ideas. Now I request Dr. P.K. Polai, KANAK Institute of Pharmaceutical Sciences, ROURKELA, for chairing the session. Over to Dr. P.K. Polai, sir. Sir, please.
Good afternoon. Good afternoon.
Sir, you have muted. Please unmute.
Good afternoon. Our next participant is Niluram Palda, sir, Assistant Professor of KANAK Institute of Pharmaceutical Sciences. Sir, please share your slide.
Thank you, Dr. Prakash Polai, sir. Sir, it is audible?
Yeah, audible.
So, very good afternoon, all of you. I am going to present my paper on soil quality in and around pharmaceutical industries. Soil quality in and around pharmaceutical industry. So, soil quality closely related to soil productivity. As we know, due to rapid industrialization, urbanization have led to degradation of our natural resource like water, soil and air. Soil resource is the important natural resource Without soil, survival of life is not possible. But after industrialization, soil nutrients quality has been affected. So many major and minor pharmaceutical industry in our country, which directly or indirectly affected the quality of a soil. Mainly, the effluents discharged in the vicinity of pharma industries and improper disposal of industrial waste on land. These are the two mainly. So the commonly used chemicals in pharmaceutical industry are titanium dioxide. As you know, titanium dioxide is a coloring agent, aromatic chemicals. So generally it is used for flavoring agents. Magnesium hydroxide, it is generally used for antacids. Similarly, organic and some inorganic chemicals, emulsifiers, charcoals, volatile oils, steric ammonium nitrates, thiamines, 3-chloropropanophenols. So these are the common chemicals which are used in the industry, which affect both physical and chemical parameter of soils like pH, water holding capacity, density, color, sodium, potassium, calcium, magnesium, sulfates, phosphates. So it is observed that some of the drugs like non-steroidal anti-inflammatory drugs, analgesic drugs, antihistaminic drugs, which contain some heavy metals like lead, cadmium, nickel, mercury, copper, which affect the soil quality like its pH, its density, its organic matter, inorganic cation and anions. And sometimes the soil biological property also affects. And also it affects some enzyme activity for plant metabolism, like the enzymes oxidase, peroxidase. This non-steroid anti-inflammatory drugs. One common example you can say antibiotic amoxicillin. So which an adsorption in soil make the soil acidic pH. So it need the pharmaceutical industry should reduce the discharge of pollutants and follow the environmental plans, and also to need a regular monitoring of concentration of pharmaceutical compounds in the pharmaceutical effluents. Thank you, sir.
Thank you, sir, for your nice presentation.
Thank you, sir.
Our next contestant is Subhanarayan Mahapatra, sir. Subhanarayan Mahapatra, sir, research scholar of BPUT Odisha.
Yes, sir.
Sir, share your slide and continue.
Yes. এটি ক্লিক করি
বললাম তো আপনার এটা করলাম
Good afternoon, madam. Sorry to interrupt. Monika, madam, good afternoon. Hello, madam.
Thank you, Prakash, sir.
Yeah.
Hello.
Start anything.
Am I audible?
Yeah.
Thank you, Prakash, sir. Yeah, this is Subhanarayan Mahapatra, research scholar of Patnaik Technical University under the guidance of Professor Dr. Mrs. Subhasri Sahoo and co-guidance of Dr. Professor Chandrakanti Chakravarti, sir. And I want to extend my best wishes to everyone here, the incredible personalities in this international conference. Given the topic for the presentation is exploitation of endocytosis and intracellular trafficking of nanomedicines. So what exactly the endocytosis? Basic understanding of endocytosis is endo stands for inside the cell, and cytosis, this is nothing but the ingestion of materials inside the cell. So once any material, any drugs, any nutrients which comes inside the body, it travels through the body and comes into the extracellular fluid, and then it crosses the plasma membrane, comes into the inside the cell. And there is a complex mechanism behind it, what we call endocytosis. So simply, the endomembrane, the plasma membrane that invaginates inside and forms vesicle and vascular transport arteries. So broadly, we can classify this endocytic mechanism into two categories. One is phagocytosis, and the second one is the gynocytosis. Micromolecules, the solid ones, normally phagocytose inside the cell, and the pinocytosis again can be subdivided into different categories like clathrin-mediated endocytosis, clathrin-independent endocytosis like caveolin-mediated. Sometimes it is independent of the clathrin and caveolin-independent. And sometimes the process which is called as macropinocytosis, the bulk amount of the liquid and dissolved materials are getting into the cell. Further, clathrin and caveolin independent pathway can be subclassified depending on the membrane-bound proteins which have facilitated for the pushing up this drug material inside the cell. ARF6 dependent, sometimes flotillin, CDC42, the cell cycle dependent controlling agents, and the ROA dependent endocytosis. And the principal components after endocytosis occur are the primary vesicular formation or the endosome formation. Next, it matures to form late endosomes. And next, as it moves on inside the cell, fuses with the lysosome and forms the lysosome and acidic environment occurs, the protein pumps gets activated, pH gets responsive towards the cell degradation, the molecular degradation, and ultimately the drug comes into the targeted area. So here is the classification on the screen, and the mechanism what I'm showing, if you can see, endocytosis, phagocytosis, macropinocytosis, clathrin dependent, sometimes it is independent, passing through the ARF6, CDC42, dynamine, intersectin, amplifies so many adapter proteins in the plasma membrane that facilitate going through this pathway. If you can see here in the screen, the clathrin independent carriers, PLIC, goes to the GAC. Next, sometimes it is bypassing the lysosome in order to avoid certain drugs which are acid sensitive, and we are trying to protect from this lysosomal degradation. So endolysosomal pathway is normally bypassed by this process. So here are the sometimes the endosome gets measured and fused with the multivascular bodies forming, and most of the Golgi, trans-Golgi networks fuses with this, and there will be intracellular trafficking, which causes the reversible and unidirectional, sometimes bidirectional movement between the endoplasmic reticulum and the Golgi bodies for packaging, repackaging, and other chemical reactions. Now here, I want to brief about phagocytosis, what exactly the mechanism behind it. Once the chip molecules or the nanocargos that we're targeting comes on the surface of the cell, first the cell recognizes in the extracellular fluid. And once it recognizes, sometimes incredible phenomena that we call opsonization that occurs, IgM, IgG, the immunoglobulins along with the complement components attaches with this and forms different complexes, what we call opsonized particles, and later they get into the cells, invaginated. There will be a pit formation, the coated plate, sometimes clathrin mediated. In case of pinocytosis, they forms the vesicle and gets in. It engulfs entire the lipid protein, what of the molecules into the vesicular form, and there will be the inward movement, and there is a pinching of the inward membrane, and they form the vesicle. Later on, the primary endosome. Next, on the maturation, they form more acidic late endosome, and later on fuses to lysosome for the degradation. And of course, after inside the cell, after the intracellular trafficking, actin, the cytoskeleton, it plays a vital role over here. And because of the spatial arrangement of this actin, and along with the amphiphysin, dynein, dynamine, so many adaptive proteins, they help in the invagination and forming the vesicles. And finally, through this process, there is a starting at the receptor ligand interaction, signaling cascade, as we know in the pharmacodynamics. There is what do we say, HSCP pathway and IP3 DAG pathway, so many transducer mechanisms, and as a part of that, it acts on, and there is an ingestion of the entire material phagosome forms in the phagocytosis. In pinocytosis, it's a complex. If you consider the classical concept of clathrin modified, clathrin mediated, and sometimes it is independent, and the membrane bound LDL, low density lipoproteins, cholesterol, transparent, it helps in the receptor mechanism and inward movement of this sorting mechanism, you can say, sorting in of these materials. And finally, this shows the action or the pharmacological action in the target areas. So considering the pharmacological effect and physiological effect, physical and chemical properties of the nanoparticles, we have to consider so many physical chemical properties during the nanoformulation. Now, coming to certain points of the endocytosis and intracellular trafficking, actin, microtubules, intermediate filaments are the driving components, and intracellularly, the endoplasmic reticulum, mitochondria, lysosome, these are the incredible cellular organelles which plays a vital role for sorting, budding, and finally, clathrin detaches and the vesicular formation docking on the plasma membrane fusion with the acceptor membrane occurs. There is two proteins on the slide. If you see this one, COP1 and COP2, these are clathrin coated proteins. Sometimes bidirectional, Golgi to endoplasmic reticulum and endoplasmic to the Golgi. This is unidirectional. Snare protein, of course, plays a vital role again for the fusion of the vesicles. Next, considering the parameters influencing the endocytosis of nanomedicine. As we know, there are certain strong interactions, sometimes ionic, metallic, covalent bonds in the preparation of the quantum dots, gold nanoparticles, carbon nanotubes, and sometimes soft nano formulation cargoes with the weak interactions like hydrophobic effect, Coulombic effect, and hydrogen bonds. So all the way they have interfacial process and interactions. Sometimes the product is targeted for to be changed under the lysosomal processing, and as it undergoes the lysosomal processing, the acidic hydrolytic enzymes degrade it and make it sensitive. But if there is acid sensitive drugs, we are going to bypass this lysosome. These are the different deconstruction endocytotic processes that enables us to formulate a better way the nano formulation and optimizing our nano product. So of course we need to have a quantitative designing and endocytic study methods. Unless until you chase in vitro and in vivo how this drug, this nanoparticle is moving and endocytic pathway, how it reacts with this, we cannot able to design a better nano formulation. So physical chemical properties, if you're considering size, shape, surface charge, hydrophobicity, so many things are pretty important taking the consideration. Say for example, if your size consider less than 5 nanometers, sometimes it is passing through the renal clearance, so it's not suitable. Even sometimes clathrin cavity mediated, then we have to check within the 20 to 40 nanometer or sometimes up to 1 micrometer under the macropinocytosis. And shape, if you consider this normally cylindrical and spherical are have a greater efficiency for the cellular uptake. Surface charge, cationics are more preferable because they have a strong electrostatic interaction with the anionic membranes. Even sometimes exceptionally anionic ions, the core with the micelles polymeric designs also are feasible. And hydrophobicity, of course, the hydrophobization of the drugs increases the most cellular uptake. But if you need a longer duration, longer circulation, then we can isolation, PVP, and so many hydrophilic polymers can be coated on this, and the modification of the design can help us, can enable us to fix the design effect. Now, endocytic study methods, we consider so many things like, LDL transparent are more prominently or predominantly found in cell, and which are acting as a fluorescent agent or the markers. Sometimes some inhibitors like chlorpromazine, and in case of cavity dependent, sometimes amyloid, different drugs markers are used to chase the nanoparticles, how it is moving in the body inside in vivo, and how it affects our reticular endothelial systems, our cell tissue inside, and giving the effect of cross-cellular uptake. So all the way, we need this surface charge, size, shape, material composition, hydrophobicity, ionization potential at the utmost precautions judiciously we have to think upon it. And coming to the conclusion part, deconstructing the endocytic pathway and physiochemical properties of nanoformulations can enable us the integrated approach of nanomolecules, nanocargos design. So these are the few things here I've written. Proteins, recombinant DNA, molecular compounds can be delivered, and preclinical trials have been conducted over here. So that is how we can effectively approach for the site-specific delivery. Sometimes the confocal microscopy and atomic force microscopy that helps for studying in detail of this size, shape, surface charge, so many things, so that you can even sometimes X-ray photoelectron spectroscopy helps us for studying the physical chemical properties and come to in granular details of these nanoparticles, so that we can help designing better nanoformulation in the pharmaceutical field for the human welfare. So this is all about. Thank you very much.
Nice presentation, sir. Our next
Thank you, sir.
is Smita Rautrai of KANAK MANJARI Institute of Pharmaceutical Sciences, Assistant Professor, Smita Rautrai.
Good afternoon, sir.
Good afternoon.
Good afternoon to everybody. Today I am going to present my slide, Role of Natural Products in Healthcare.
Madam, slide. Madam, share your slide.
Yes, sir. Sir, is it visible?
No, ma'am. Yeah, it is visible right now. Yeah, visible.
Yes, sir. So, good afternoon, everybody. Today I am going to present Role of Natural Products in Healthcare. As we all know that natural products plays an important role in our daily life. Natural products discovered to deliver many layered compounds in therapeutic treatment. Every plant is identified its own medical properties. The therapeutic role is due to bioactive molecules that are more effective and less toxic. For example, discovery of antibiotic penicillin is from fungus Penicillium notatum, which is a natural product. Another example is licorice root, that is used in the treatment or prevention of disease like HIV, hepatitis, and influenza. So there are many steps. There are many steps that medicinal plants have good therapeutic properties. Plants are used as a direct sources of therapeutic and bioactive agents. Bioactive fractions serves as raw material. The isolated structures derived from plant species can be used as a lead for new discovery of drugs. So plants can be used as a bioactive markers for spectroscopic and chromatographic analysis for new discovery of drugs from natural resources. Isolation, identification, and characterization of phytochemicals can be usually present in different, can be extracted from different plant extractions. The bioactive compounds have different properties, separations, identification, and characterization of bioactive compounds. Phytochemical screening assay is simple, quick, and inexpensive procedures. So these are the steps to get the lead of the natural products. Lastly, I concluded with the aim that in near future, natural products are a great boon to the world for existence. Moreover, natural products will be important sources of new pharmaceutical companies. Thus, the world is always gifted with the nature and man is gifted with brain. So, let us make use of discovery of new entities that are available for common people in the economical rate, and you will be happy to lead a life in the earth. So in the new era of 21st century, no life is possible on earth without herbal drugs or products that are obtained through natural herbal drug discovery. So it's a credit to our earth. We are having natural products. So that's what. Thank you, sir. Thank you, everybody.
Thank you, madam. Nice presentation. So next participant is Asant Malik of KANAK Manjari Institute of Pharmaceutical Sciences. Asant Malik. Asant Malik. Sorry, he is not present. So over to Subhasri ma'am.
Thank you, sir.
Thank you.
Now I request Professor Dr. Biswa Ranjan Behera sir, Principal KANAK Manjari Institute of Pharmaceutical Sciences, ROURKELA, to give welcome address to Dr. Monika Machoy, madam, as the keynote speaker. Over to sir.
Good afternoon to all, and once again welcome you all to this international conference on New Horizon in Pharmaceutical Research. Before I start the introduction of Dr. Monika Machoy, let me confirm that okay, she has joined. Thank you, madam. Am I audible, madam?
Thank you very much. I would like to thank.
Okay, let me introduce. First of all, let me introduce you before starting your keynote address. So, Professor Dr. Monika Machoy, MD, graduated with distinction from the Faculty of Medicine and Dentistry at the Pomeranian Medical University in 2009. Defended her PhD thesis with summa cum laude in 2016. And is currently employed as a researcher at the Department of Orthodontics of the Pomeranian Medical University. She expanded her professional knowledge during 1 year of scholarship at the Ernst Moritz at the University of Szczecin, Germany, and during a three-year internship at the Technical University in Dresden, Germany. She has been a speaker at numerous orthodontic conferences, among others American Association of Orthodontics, European Orthodontic Symposium, and numerous conferences of the Polish Orthodontic Society. She conducts research focusing on the effect of orthodontic treatment on dental and periodontal tissues and the cause of pain in orthodontic patients. She has so far authored more than 25 publications. To her credit, she has presented her research work at 12 national and international conferences and is the member of 2 scientific societies. So, Dr. Monika Machoy, madam, will be delivering a keynote address on the topic Advances and New Trends in Dental Pharmacotherapy. So, welcome, madam.
Thank you very much for the invitation. It is an honor for me. And the topic of my presentation are advances in dental pharmacology, but I would like to focus on silver nanoparticles in dental treatment. I would like to give you my presentation to share it with you. Just wait a minute, please. Can you see my presentation now?
Yes, ma'am. Yes, yes.
Great. Super. Okay, great. Advances and new trends in dental pharmacotherapy. I would like to focus on silver in dentistry in silver nanoparticles. The use of silver in oral care has been known for centuries and gained worldwide spread in the 19th century as the main components in dental amalgams used for tooth restoration. Its use in amalgams has been reduced in 1930 as they were progressively substituted by aesthetic polymer-based resins. Since nanoscience has evolved and the outstanding antimicrobial properties of nanostructured silver-based formulations have been demonstrated against microorganisms such as bacteria, viruses, and fungi, the interest in silver has been renewed, and several promising new technologies are currently under development, especially in dental materials. In this context, silver nanoparticles have been demonstrated to be effective antimicrobial components in prosthetic materials, adhesives, and implants to promote caries arrestment, to prevent biofilm formation, and for osteogenic induction. Metallic silver is rather unreactive. On the other hand, silver has the lowest first ionization potential in group 11, and thus silver cation is the most stable species in aqueous solutions, including physiological fluids and its solids. Due to the filled third orbital, silver cation compounds present a significant covalent character, and they do not tend to form organometallic compounds. In addition, they do form complexes with coordination numbers as low as 2 silver ions from bonds, preferably with sulfur, but also with nitrogen and oxygen. So silver cation can bind to enzymes with S-bending groups, as well as N atoms from nucleic acids. Besides formulations containing silver cation, other silver-based compounds have also been successfully applied in dentistry. For instance, the linear complex, which was recently cleared for caries arrestment in the US, is used in Japan for more than 80 years. However, with the development of nanotechnologies, silver nanoparticles have gained most of the focus. It is now possible to produce silver nanoparticles with controlled size and morphology, high homogeneity, in other words, low polydispersity index, and specific target functions. In other words, functionalized with molecular capping agents from small hydrophilic and hydrophobic chemical groups to large biomacromolecules such as proteins. The nucleation and growth process of silver nanoparticles can be mediated both by synthetic reagents and by biologically available products from plants and microbes. As the nanoparticle composition, particle size distribution, morphology, and surface chemistry can be finely tuned. Silver nanoparticles can access different sites in the oral cavity. In such a way, they can currently be conceived as multifunctional building blocks for dental materials and dentistry protocols. Although the antibacterial mechanism of silver nanoparticles has not been fully elucidated, some aspects of the antimicrobial action of silver nanoparticles have been recognized. Silver ions are capable of acting on different structures of the bacterial cell. Primarily, these ions seem to adhere to the cell wall and cytoplasmic membrane through electrostatic attraction and affinity to sulfur proteins, thus enhancing the permeability of the membrane and also leading to disruption of the structures. In gram-negative bacteria, porins in the outer membrane are also involved in the uptake of silver nanoparticles. Bacterial molecules that can be damaged by silver nanoparticles include DNA, proteins, and lipids. Silver nanoparticles also stimulate oxidative stress response, causing bacterial cell destruction, and increase the phosphorylation of tyrosine reduces on bacterial peptide substrates, inhibiting bacterial growth and viability. Cell membrane disruption and DNA modification via reactive oxygen species as the principal agent were also described in the literature. Bacteria in the oral cavity are preferably organized in biofilms, which confer better conditions for growth, immunological evasion, and resistance to antibiotics. Specifically in dentistry, the preparation of nanoparticles must take into account the biofilm architecture and the mechanistic aspects of silver nanoparticles. The nanoparticle's properties may affect its efficiency and interfere with its mechanism of action. Important aspects to be mentioned in this connection: the diffusion of nanoparticles in biofilm exhibits an inverse relationship between effectiveness and size. Nanoparticles over 50 nanometers are not able to penetrate the biofilm due to the relative self-diffusion coefficients in the biofilm, and this decreases exponentially with the square of the nanoparticle diameter. In addition, charged nanoparticles do not diffuse easily through the biofilm, probably because the presence of phosphoryl and carboxyl groups on the surface of the bacteria, which gives the cell surface an electronegative character. The concentration in formulations is commonly given by the total amount of silver, metallic silver, and silver cation, and this value is provided in gram to milliliters. The commonly used technique to determine the total amount of silver per milliliters is ICP, inductively coupled plasma spectrometry. In some cases, it is differentiated the metallic silver nanoparticles and the silver ions. However, this concentration is difficult to be compared because the dissolution of the silver cations and thus its concentration depends on several synthetic and processic parameters, such as the medium composition and ionic strength, silver nanoparticles size and morphology, and the silver nanoparticle scapping agents. It is currently accepted that the actual antibacterial element is silver cation, but silver nanoparticles can be oxidized inside bacteria. In this case, aspects involving the bacterial uptake, penetrability of silver nanoparticles become very relevant. It is also important to mention that the penetrability of ions and nanoparticles are different. Due to necessity of silver nanoparticles dissolution for effective antibacterial action through a silver cation release, the size and shape of the format particles can influence their action along with their concentration. Between nanoparticles with spherical or quasi-spherical format, those with smaller size have higher activity once their surface exhibits a larger area, thus being more prone to dissolution when compared to larger silver nanoparticles. This also can explain why aggregated nanoparticles possess lower antibacterial activity, since they had a smaller surface area exposed to solvent than isolated nanoparticles. It was recently presented that the activity of silver nanoparticles with sizes less than 10 nanometers is due mainly to the nanoparticle itself, while for those larger than 10 nanometers, the predominant mechanism occurs through the silver ions. In this way, the precise range of size in which silver nanoparticles have antimicrobial effects is yet to be determined. Furthermore, it is important to research how the silver nanoparticles are oxidized to silver ions in the cells. Finally, but not less important, we still do not have an historical report on the long-term bacterial response towards silver nanoparticles. These studies are imperative to assess the possible bacterial resistance to silver nanoparticles. Despite all these issues, studies have shown that silver nanoparticles have considerable potential as alternatives in antibacterial applications in the future. The time necessary to completely release silver cation from silver nanoparticles depends on their dissolution process, which is attributed to oxidation of the metallic silver. The dissolution depends on the medium ionic strength, pH, concentration of the dissolved oxygen, and temperature, presence of complex zinc ligands, surface coatings, and silver nanoparticles shape and size. For instance, some researchers examined size-dependent dissolution of silver nanoparticles with different sizes, 6, 9, 13, and 17 nanometers, coated with thiol functionalized ligands, and neutral and acid pHs by measuring the silver concentration up to 80 days. They demonstrated a size-dependent dissolution increased as the size decreased in both solutions and a faster dissolution in the acidic solution. Moreover, transmission electron microscope images demonstrated that the particle morphology was not affected by this solution and no aggregation was observed. Clinical applications of silver nanoparticles in dentistry. Research on silver nanoparticles has become an emerging field in recent years. The first reason for this is that silver nanoparticles can be synthesized by mediating their nucleation and growth process using different synthetic reagents. Second, silver nanoparticles can be specifically functionalized with molecular capping agents such as proteins and chemical groups. Third, silver nanoparticles possess a strong antibacterial effect that improves clinical treatment outcomes. Therefore, homogeneous silver nanoparticles with controlled size, morphology, and function can be regarded as multifunctional building blocks in diverse dental materials. Silver nanoparticles can be added to acrylic resins for fabrication of removable dentures in prosthetic treatment, composite resin for direct restoration, irrigating solution and obturation material in endodontic treatment, adhesive materials in orthodontic treatment, membrane for guided tissue regeneration in periodontal treatment, and titanium coating in dental implant treatment. Acrylic resin is commonly used to fabricate removable dentures. Opportunistic oral pathogens can colonize acrylic materials, causing dental infections such as denture stomatitis. Silver nanoparticles can be added to acrylic resin to inhibit the growth of such bacteria as Streptococcus mutans, Escherichia coli, and Staphylococcus aureus. In addition to the antibacterial effects, acrylic resin incorporated with silver nanoparticles has also displayed antifungal properties against the adhesion of Candida albicans, which is one of the key opportunistic pathogens on the denture base. Silver nanoparticles can also enhance the mechanical properties of acrylic resin. The effect of silver nanoparticles on the mechanical properties is influenced by concentration of silver nanoparticles, the type of acrylic resin, and the polar interactions between the polymetal methacrylate chains and silver nanoparticles. Silver nanoparticles can increase the flexural strength and elastic modulus of acrylic resin. Moreover, they can increase the thermal conductivity and compressive strength of the acrylic base. Silver nanoparticles in restoration treatment. Residual bacteria in the prepared tooth cavity and colonized bacteria in microleakages along tooth restoration interfaces may result in secondary caries of the dental restoration. Silver nanoparticles can be added to adhesive systems and composite resins. They can prevent secondary caries by bringing notable antimicrobial properties to bear at low concentration. Restorative adhesives containing silver nanoparticles can interfere with biofilm formation due to the widespread inhibitory action of carcinogenic bacteria. Composite resin with silver nanoparticles had no significant adverse effect on fibroblasts because the silver ions released per day are very low. In addition, researchers immobilized silver nanoparticles in a biocompatible film of polymers to obtain a maximum lethal effect on microbial cells without killing human cells. Silver nanoparticles can also be added to porcelain to enhance its mechanical properties. Porcelain's reinforced mechanical properties, such as hardness and fracture toughness, can be attributed to the residual compressive stress generated by an ion exchange reaction and differential thermal expansion of silver nanoparticles. Silver nanoparticles in endodontic treatment can be used as a substitute of sodium hypochlorite in intracanal irrigation for endodontic treatment. Gutta perca coated with silver nanoparticles has been developed as an antimicrobial obturator for root canal obturation. Silver nanoparticles are also incorporated as antibacterial material into mineral trioxide aggregate to enhance the success of pulp capping, apexification, and sealing perforations in teeth. Sodium hypochlorite is regarded as the gold standard for chemical disinfection of root canals in endodontic treatment. However, sodium hypochlorite reduces the elastic modulus and flexural strength of dentin and causes toxic damage to the periapical tissues. A study found that root canal irrigation with silver nanoparticle solution did not significantly affect the mechanical properties of dentin. Silver nanoparticles in endodontic treatment. Another study found that a low concentration of silver nanoparticles had superior biocompatibility relative to sodium hypochlorite. In addition, silver nanoparticle-based irrigation solution was as potent as sodium hypochlorite in the elimination of both Enterococcus fecalis and Staphylococcus aureus. Therefore, some have suggested using silver nanoparticle solution for root canal irrigation during endodontic treatment. Gutta perca is commonly used in root canal obturation, although it is desirable that endodontic obturators have antibacterial properties. Gutta-perca's antibacterial properties are limited. A study found gutta-perca coated with silver nanoparticles had antibacterial and antifungal properties and was as effective as conventional gutta-perca in preventing bacterial leakage. In addition, there is no difference in the cytotoxicity of fibroblasts and subcutaneous tissue reaction or inflammation between gutta-perca coated silver nanoparticles and conventional gutta-perca. Mineral trioxide aggregate has extensive applications in dentistry, such as pulp capping, apexification, and sealing perforations. It is essential to add antimicrobial agents into mineral trioxide aggregate to enhance its antimicrobial properties. Mineral trioxide aggregate combined with silver nanoparticles can enhance antibacterial activity against anaerobic endodontic pathogens. The antifungal ability against Candida albicans was also improved by the addition of silver nanoparticles to mineral trioxide aggregate. Moreover, research has reported that mineral trioxide aggregate incorporated with silver nanoparticles can increase pH value and compressive strength with proper radiopacity and shorter setting time. In addition, researchers have revealed that mineral trioxide aggregate with silver nanoparticles has good biocompatibility without inducing inflammatory reaction. Silver nanoparticles in orthodontic treatment. Silver nanoparticles can be used to prevent enamel caries, so-called white spot lesion, which is a common complication for patients with orthodontic treatment. Silver nanoparticles can be added to adhesive materials such as resin-modified glass ionomers and composite adhesives, but inhibit biofilm growth. Orthodontic elastomeric modules such as ligatures can be applied and allied with silver and nanoparticles to prevent enamel caries. They can affect the adhesion of Streptococcus mutans to the materials by contact with bacteria and inhibit bacterial action around brackets and wires by the release of silver ions. The materials can release silver ions for 4 months to exert a long-term antibacterial effect. Materials with silver nanoparticles are biocompatible and have no significant cytotoxic or mutagenic effects. Moreover, the materials have negligible irritation and delayed type hypersensitivity potential. There are no adverse effects on their mechanical properties, such as shear bond strength. Dental implant treatment with nanosilver. Peri-implant infection is a major threat for implant treatment. Conventional antibacterial methods are insufficient due to constant risk of infection from peri-operation, which persists until later in life. Modifying implant surfaces with silver nanoparticles using various doped methods is currently an area of intensive interest due to the safety and strong antibacterial properties of silver nanoparticles. Researchers have demonstrated the long-term antibacterial effect of titanium implants coated with silver nanoparticles by steadily controlling silver ion release with various coating methods. The antibacterial effect can also be prolonged by immobilizing silver nanoparticles on implant surfaces.
The embed silver nanoparticles inhibit bacteria without consumption through continuous direct contact with bacterial cells. Although decreasing the size of silver nanoparticles generally increases their antimicrobial ability, larger size silver nanoparticles embedded in titanium had a better antibacterial effect due to the consumption of more protons. Silver nanoparticles can kill Staphylococcus aureus and Pseudomonas aeruginosa at a low concentration, which has no significant cytotoxic effects on osteoblastic cells. In addition, titanium embedded with silver nanoparticles can enhance bone mineral density, bone formation, and trabecular pattern with no harm to tissues adjacent to dental implants. Silver nanoparticles in periodontal treatment. Periodontitis is a prevalent chronic inflammatory disease caused by various types of microorganisms. Adequate infection control is essential for periodontal treatment through disruption of the biofilm and suppression of inflammation. Compared to traditional antibiotics, silver nanoparticles possess antibacterial properties without the generation of bacterial resistance. Silver nanoparticles significantly enhance bactericidal properties when combined with antibiotics, including cefotaxime, ceftazidime, meropenem, and ciprofloxacin. Importantly, inactive antibiotics restore strong antibacterial activity against multi-resistant bacterial strains when they are combined with silver nanoparticles. Silver nanoparticles synthesized with an appropriated capping agent can promote the inhibition effect against gram-negative bacteria that predominantly cause periodontal infections. Smaller size silver nanoparticles presented a higher antibacterial property against oral anaerobic pathogenic bacteria. Because guided tissue regeneration membrane with silver nanoparticles reduced adherence and penetration of bacteria, treatment of intra-bony defects using guided tissue regeneration membrane with silver nanoparticles can improve clinical success. Silver nanoparticles were reported to possess an anti-inflammatory effect through modulation of the level of inflammatory cytokines and growth factors. Periodontal dressing coated with silver nanoparticles can be used to treat gingival wounds. This treatment accelerates the early phases of wound healing with apparently new collagen synthesis and neovascularization. However, some silver nanoparticles can provoke inflammation and foreign body reaction in periodontal dressing. Is it safe or is it toxic? Some researchers are concerned about the toxicity of silver nanoparticles. The toxicity of silver nanoparticles is directly associated with the free silver ions. Because of the nanoscale size of silver nanoparticles, they can readily disturb biological molecules, cells, and human organs. Some laboratory studies have reported that silver nanoparticles can induce oxidative stress and impair mitochondrial function of human cells. Moreover, silver can be detected in organs after administration of massive silver nanoparticle doses, especially in the liver and spleen. Researchers are also concerned about the ability of silver nanoparticles to cross the blood-brain barrier via transsynaptic transport and accumulate in the brain. However, even at low concentration, small-sized silver nanoparticles decrease some inflammatory cytokines and angiogenesis parameters. Silver nanoparticles can express anti-inflammatory properties. They are biocompatible with fibroblasts and keratinocytes. Silver accumulated in organs can mostly be cleared after 8 weeks. An animal study using rats found no observable adverse effect with the oral administration of silver nanoparticles. Another clinical study did not find clinically important observable toxic effects on patients taking commercial colloidal silver products. Researchers discovered mineral trioxide aggregate with silver nanoparticles did not induce a significant inflammatory reaction on subcutaneous tissues of rats. It has also been proven that periodontal dressing with a high concentration of silver nanoparticles is biocompatible with gingival wound healing. Although authorities have not fully acknowledged the safety of silver nanoparticles, clinicians often apply silver nanoparticle-based wound dressings to their patients' wounded skin. Silver nanoparticles can enter into the body through a wound because the strict barrier is lost. These silver nanoparticles are phagocytosed by macrophages and may cause immune system perturbations. Silver nanoparticles also enter other organs, such as the liver and spleen, via circulation. Nevertheless, this systemic toxicity of ingested silver nanoparticles has not been reported. Another concern is their potential hazard to the marine life if they are released into the environment. However, the toxicity of silver nanoparticles may be increased or reduced when they interact with materials and organic compounds. Hence, nanoparticles can have a harmful or helpful effect to the environment. It is essential to perform a nanomaterial-specific evaluation to ensure their use is safe for humans and the environment. Conclusions. Incorporating silver nanoparticles into dental materials may enhance the mechanical features and antibacterial properties of the dental materials. Although the mechanism of silver nanoparticles' antibacterial effects is not yet fully understood, many researchers believe that silver nanoparticles can continually release silver ions to kill microbes. An increasing number of dental materials with silver nanoparticles are being developed for prosthetic, restorative, endodontic, orthodontic, periodontal, and implant treatment. Some laboratory studies reported that silver nanoparticles have cytotoxic effects on human cells. However, the clinical significance of the potential toxicity of silver nanoparticles remains unknown. Further studies are essential because clinical evidence is still limited. Thank you for your attention, and I invite you to cooperation. Thank you very much.
Thank you very much, madam. Thank you, Dr. Machoy, for informing all participants about silver nanoparticles in dental pharmacotherapy. It was a very nice and informative session. I, on behalf of KANAK MANJARI Institute of Pharmaceutical Sciences, ROURKELA, India, extend my deep gratitude for sparing your valuable time with us. Hope this may continue in future. Thank you very much. Thank you very much, madam.
Thank you. It was a pleasure. Thank you.
And now I hand over to Dr. Subhashree Sahoo for the next session. Dr. Subhashree Sahoo. Dr. Subhashree Sahoo.
Yes, sir. Thank you very much, madam. Thank you very much, sir. Now I request, this is a very nice and informative session about the silver nanoparticles. Now I request our principal, sir, Professor Dr. Biswa Behera, sir, to felicitate Dr. Monika Machoy.
Thank you, madam.
Thank you very much.
Now, on behalf of KANAK Manjari Institute of Pharmaceutical Sciences, ROURKELA, would like to request Dr. Monika Machoy, madam, to receive a certificate of honor as a token of love and affection for being a valuable resource person on this conference. Thank you, madam. Please, Subhashree, madam, please share the certificate so that.
Thank you.
Please accept this as a token of affection, madam. Please be on the line. Subhashree, madam, can you share? Subhashree, madam, can you share?
Yes, of course, of course.
Please be online, ma'am. Please be online. Subhashree,
Yes,
madam.
yes. Okay, thank you.
Subhashree, madam.
First, madam has to stop the sharing of the slides. Then only I can present the slide.
Madam, Michael, madam, please stop sharing your slide so that we will share the certificate.
For sharing the certificate, our chairman, sir, Dr. Biswa Patnaik, sir, now interested to interact with madam.
Professor Machoy, thank you for your delivery.
Thank you very much.
Yeah, we have enjoyed it very much. So we would like to show our respect by honoring you through certificate.
Thank you.
Please accept it.
Thank you very much. It's an honor for me.
Machoy, please accept the certificate of honor.
Thank you very much. Wow, it's great. It's so beautiful. Thank you.
Can you come to India if you are invited?
Oh, that will be great. When the COVID is over, I will come. Thank you. And we invite you cordially to Poland. It will be great to see you here. And if you could be our guest of honor. Thank you.
Thank you, madam.
Thank you very much.
Actually, still now, Professor Dr. Pravat Kumar Sahoo has not joined. So now, can I request any of our members, those who are interested to present, they can present and share their views and ideas. Can I request Subhanaran Mahapatra sir to sing a song? Am I audible, Subha sir?
Yes, ma'am. Hello. Am I audible? Hello?
Yes, yes, sir.
I am going to sing a Hindi song. It's from old movie and sang by Kishore Kumar. प्यार दीवाना होता है मस्ताना होता है हर खुशी से हर गम से बेगाना होता है प्यार दीवाना होता है मस्ताना होता है हर खुशी से हर गम से बेगाना होता है समा कहे परवाने से परे चला जा मेरी तरह चल जाएगा यहाँ नहीं आ वो नहीं सुनता उसको जल जाना होता है हर खुशी से हर गम से बेगाना होता है प्यार दीवाना होता है मस्ताना होता है हर खुशी से हर गम से बेगाना होता है रहे कोई सौ पर्दों में डरे शर्म से नजर अजी लाख चुराए कोई सनम से आ ही चाहता है जिस पे दिल आना होता है हर खुशी से हर गम से बेगाना होता है प्यार दीवाना होता है मस्ताना होता है हर खुशी से हर गम से बेगाना होता है थैंक यू
Thank you very much, sir. Now I request Dr. Manoj Kumar Mahapatra sir, KANAK MANJARI Institute of Pharmaceutical Sciences, ROURKELA, to welcome our esteemed guest of the evening, Professor Dr. Pravat Kumar Sahoo sir, Director DIPSAR, New Delhi. Hello, Manoj sir. Hello. Manoj sir. Am I audible, Manoj sir?
Madam, yes.
Manoj sir, please welcome our esteemed guest of the evening.
Thank you, madam. I welcome you all to this afternoon session of International Conference on New Horizons in Pharmaceutical Research. And it's my pleasure to welcome Professor Pravat Kumar Sahoo, sir.
Manoj sir, please raise the sound of your system.
Okay. Am I audible now, madam?
Yes, now audible.
Okay. Okay. Good evening, all. And it's my pleasure to handle this afternoon session to welcome Professor Pravat Kumar Sahoo, sir, an eminent speaker and currently holding the post of Director, DIPSAR, New Delhi, who will be addressing us regarding NEP, that is National Education Policy 2020. So before inviting Professor Pravat Kumar Sahoo, sir, please allow me to read a short brief about his lifetime achievements. Professor Pravat Kumar Sahoo is currently holding the post of Director, Delhi Institute of Pharmaceutical Sciences and Research. He had pursued his M.Pharm as well as PhD from Andhra University and completed MBA from Xavier's Institute of Management. In his 35 years of professional tenure, he has immense contribution in academic, research as well as administrative fields. He started his career as manufacturing chemist in pharmaceutical industry. Then he joined academics as a senior lecturer in Sri Jaydev College of Pharmaceutical Sciences and Research. He has served as regional director of AICTE and later on rose to the position of director AICTE. He joined DIPSAR as a professor in 2004. His academic and research credentials include more than 50 research and review articles, many book chapters, and a complete book on pharmaceutical marketing. He has guided over 60 master students and 6 PhD students. He has been appointed as an expert member in various statutory bodies like PCI, AICTE, NTA, GPAT, APTI, Delhi State Branch, and Association of Indian Universities. He has compiled study materials for continuing education program of AICTE and also developed the modern syllabus for pharmacy education. He also actively played the role of resource person and delivered many invited talks in hundreds of seminars, conferences, as well as workshops. So with this, I would like to request Professor Pravat Kumar Sahoo, sir, to please address the audience. Please, sir. Sir, please unmute yourself.
Please unmute, sir. Sir, please unmute.
Thank you, Dr. Manoj, for giving a nice introduction. Actually, today I will give a very short presentation on national education policy. Actually, so somewhat I'm not feeling well, so that was the reason I asked Professor Subhasri to cut down my lecture to only 10-15 minutes. So I have joined in this afternoon for that. So my topic will be National Education Policy 2020. And this is this education policy has come into effect after 34 years. The last education policy was in 1986. And you will know that all these education policies were framed by Government of India. It was during the Congress government in those periods, and mostly the education policy and the educational framework, whatever, it was away from this Indian culture, mostly the different religious teachings. Those were left out, and Indian culture also, it was not much reflected in those education policies, and people think that by adopting the Western culture and the Western education policy was it will be helpful. But however, during this new national education policy, Professor Kasturi Rangan, the famous educationist, he has made this national education policy, where in the school and elementary education level, the Indian culture, Indian learnings, teachings, everything has been inculcated in this policy, so that will not forget our own cultures and the values which has been taught in our great Ramayana or Mahabharata, whatever it may be, that is also there. So we'll not forget our own culture. That is in the elementary and the school education level, those things has been included. The local languages and Indian languages has been given more importance in this elementary education and this school education level. And the pattern has been also changed, education pattern. The same 15 years of education, but it is divided into 6, 3 plus 3 plus 3. That way they have modified. Coming to the higher education, the government of India thought that there are so many education regulatory authorities in India. So to remove the difficulties of these different multiple educational authorities, multiple acts, multiple laws regulating the higher education, the government of India has thought, no, there will be a single education regulatory body in case of higher education. That is called NHERA, National Higher Education Regulatory Authority. That is going to come into effect. And under this National Higher Education Regulatory Authority, all the different professional bodies like All India Council for Technical Education for Engineering, Pharmacy Council of India for Pharmacy, Medical Council of India for Medical, Dental Council for Dental Nursing and other bodies, they will come under one umbrella, that is National Higher Education Regulatory Authority, and UGC will become a grants giving authority. That will be called National Grants Giving Authority, and there will be NRF, there will be National Research Foundation to encourage the research activities in education, higher education level. That will be National Research Foundation. And there are other bodies which will be there to regulate the quality. There is National Assessment and Accreditation Council, that is NAC. And there is another council, that is National Board of Accreditation. These two things are looking after, NBA is looking after these technical institutions and NAC is looking after the standalone institutions and all the universities. So they measure their quality standards, that is National Accreditation and Assessment Council. So all these bodies will be there to regulate the quality, and in future the NBA will be merged with this national. There will be single accreditation council, that will be National Assessment Accreditation Council, that will be effective after some time, and NBA, National Board of Accreditation, will be merged in that, and a single body will be there to give you the quality of the different technical educational institutions. Further, there will be national institution, framework will be the NIRF, National Institution Ranking Framework. That will be there. That will show the ranking of different institutions or universities in their respective fields. So these are the new things which is going to come in this National Education Policy 2020. This National Education, before going to this National Education Policy, I'll tell you what are the different type of institutions we have in our country. In our country, if you will find, go to different educational institution, you will find our country has so many number of institutions. Total number of institutes, universities, it is 927. We have that much, no, sorry, total number of universities 993 universities in India, and out of these 993 universities, 385 universities are affiliating, 16 universities are exclusively for women, and 148 general and 142 technical, 63 agricultural universities, 23 law universities, 13 Sanskrit universities, and rest language and other universities are 106. And 60.53% of colleges are located in the rural area, and 2.5% colleges they offer PhD program, and the gross enrollment ratio which is there in case of higher education, it is 26.3%, and in that the male is 26.3, and female it is 26.4%. About 79% of the students are enrolled in the undergraduate level program, and in PhD there is the number is only 0.5%. That is the PhD. And if you come to the different central universities, the other university that is also breakup, there are 46 central universities, and there is only one central open university, and national institution of national importance 127, and state public university 371. And institutions, those are specially granted under state legislature are 5, state open universities are 14. Out of that, one is there in state open university in Odisha also. And state private universities, there are 304 private state universities, and state private open university there is only one in India, and there is government deemed universities 34, and government aided deemed universities 10, and deemed university private also 80. So this gives a picture of 993 universities which are there. If we find there are central universities, so many IITs, total 23 IITs, and Indian Institute of Science and Education Research 7, NIT 31, and triple ITs, there are 25. So these are the different strength of this higher education system in India, that so much of universities and AICTE approved institutions, more than 12,000 institutions in technical education. There may be 30,000 institutions under this institution or standalone colleges under this higher education system. So this is the strength of universities. Now coming to the national education policy and how our pharmacy education is placed in that, what will be the role of our Pharmacy Council of India, that I will show you through one slide. Just a minute, I'll come to that. Whether the slide is visible? This slide is visible?
No, sir. No, sir.
No, sir.
Achha. This is not visible. Present now. Then share. Cancel. Present now. Enter screen. Then share. Why it is not visible? That I don't understand.
Sir, actually, you have minimized the file which you want to show. You have minimized it. So please try to put that on desktop. Then only you can see the file and you can select.
I have minimized, but anyhow, okay. No. Share.
Yes, sir.
Now it is visible. So this is the national education policy that it came into effect in this year, and drafted in 2019, and it came after 34 years. The last education policy was 1986, then it was modified in 92, and this is the education policy. It came this last year to education policy 2020, and work is also going on in every sphere to merge these bodies, all these bodies, into a single body that is called the national higher education. And in this national higher education policy, the different relevant chapters of our higher education, those are described in chapter 16 and chapter 18. And this chapter 16, it states that the regulators like Pharmacy Council of India, AICTE, UGC, Dental Council, Medical Council, all professional bodies. There are 17 professional bodies in India. So those will come, for example, PCI, it was framed in 1948, UGC 56, AICTE 87, and MDA, it emerged from AICTE, and NAK is also a subsidiary body framed as a society by UGC. These are the, and all these bodies will be part of this new education policy, but there, and this chapter 18 of the National Higher Education Regulatory Authority, that will come into picture, where they have mentioned that this National Higher Education Regulatory Authority, that there will be no UGC, there will be no AICTE, no PCI, no Medical Council, but there will be a body that is called National Higher Education Regulatory Authority, and all these AICTE, PCI, Medical Council, what will be there, they will be called the PSSB, that is called the Professional Standard Setting Bodies. So they will set the standards for AICTE will set the standards for engineering, PCI will set the standards for PSA pharmacy, and this Medical Council for medical education, those things they will set the standard, and that will be regulated by this National Higher Education Regulatory Authority. That will be the umbrella body of the higher education and as well as the technical education. The regulation what will be there, but that will be light, but it will be tight regulation. But they will follow the rules very strictly, and it is not that whatever criteria they will follow that is difficult to prepare. Whatever criteria they will prescribe, it will be easy criteria, not very. So that is called, it is light criteria, but those criteria has to be followed strictly. That's why it is called light criteria. Word tight means the whatever criteria will be prescribed, that has to be followed 100%. That is a light but tight regulation. And a few of the parameters to be strictly followed. One parameter out of that is called accreditation. All the institutions has to undergo accreditation. That is the policy of the Education Regulation 2020. That all the institutions gradually through it may take 2 years, 3 years, 4 years, but all the institutions will be accredited. So after accreditation, they will be able to receive different benefits from the Government of India. Or just a minute. And the accreditation will be made once every 5 years, and the accreditation will be conducted by this National Accreditation and Assessment Council, and the fee will to be very less amount. It is not very high. For example, if you go for accreditation of your pharmacy program under this NBA, it is asking more than a pharmacy degree, diploma, and MPharm, 2-3 MPharm. If you are going to accredit in NBA, it is asking about a fee of 15 lakhs to 18 lakhs you have to pay. But that system will not be there once this new regulation comes. The fee will be very minimum. It is not that 15 lakhs, 20 lakhs. Many institutions, they are unable to go for accreditation because of the high fee. And so they have just focused on this, that it should be, fee should be economical, and the standard setting has to be done. That is the purpose, that everybody, all institutions has to be accredited. And these are the bodies which will transform like this, that NAC will continue as it is. It will be National Accreditation and Assessment Council, and UGC will be transformed into HEGC, Higher Education Grants Commission. That will be the status of this UGC. And NCT, AICTE, MCI, PCI, BCI, all will be there, but they will be called as the professional standard setting body. They don't have any power to approve any institutions or grant increase in intakes, whatever, or those approving power will be taken away from all these bodies. AICTE, MCI, Pharmacy Council of India, Medical Council, that will be shifted to the National Higher Education Regulatory Authorities. They will take over this, but these bodies will be there to prescribe the standards. What should be the standard of the education of their respective profession? That will be guided by this PCI, Bar Council or Medical Council, whatever. Then the new thing, there will be a General Education Council. There will be also a National Higher Education Qualification Framework will be there, where you can carry your credits, all the learnings, if you have, it will be in the form of credits. Suppose you have covered some 10 credits or 12 credits, and total course credit is 24. If you have got 12 credits, you may get a diploma. And with that 12 credits, if you get the diploma, you can go for a job somewhere. Suppose you have any difficulty, you can go for a job. Again, when you come back, you start from the 13th credit and you complete the 24th credits, then you will get a degree. So even after 10 years, 15 years, if you are interested to get, you will not start from the beginning. All credits you will not. And if there is some common credits in some courses, suppose in engineering, there are so many credits which are common. So after doing that course, be civil, suppose in that the mechanics, your drawing, different papers, common engineering papers, those are common in mechanical engineering also. So once you complete these common credits, you can carry to the other course, and you can get a mechanical engineering also by completing the other credits in that degree. So that is the credit carrying facility. Whatever credit you earn, that will be credited in your account, just like a bank passbook. Whatever credit, suppose you have done some course, some credits in computer science, then you have a B Pharm degree or M Pharm degree, and you have done some credits in computer science. Those credits, again, suppose you are interested to have one computer degree also, these credits you need not repeat. That will be there in your account. The remaining credits you complete, you will get the Masters in Computer Science. So that way, this new education regulation, it helps. And the other bodies which will be framed under this National Higher Education Regulatory Authority, that is Rashtriya Shiksha Ayog, and there will be Rashtriya Shiksha Appointment Committee. The way people are appointed by this Union Public Service Commission for different jobs, higher jobs, that way in due course of time there will be Rashtriya Shiksha Appointment Committee. They will only appoint the to those posts of a professor, lecturer, reader, or whatever it may be. That will be through that committee. And there will be a standard setting commission. That standard setting commission, it will consist of all these professional bodies, and each body will have an independent board that is called IB, independent board. All IB has to be constituted. Rashtriya Shiksha Ayog will appoint all the independent boards. So these are the different bodies, different features which has come under this new education policy. And there is a national research foundation is going to be framed. That is that all the institutions of India are not very much well into research. So that is the reason they have compared with other countries. In USA, 2.8% of the GDP is spent on research. In China, 2.1% of the GDP is spent in research. Israel, it is a small country, but 4.3% of the GDP is spent in research. Korea, that is also a small country. And here the research expenses are very high, 4.2% of the GDP. So in India, the researchers per 1 lakh of population is only 15, but in case of China, it is 111. And in case of USA, the people, those who do research, 425, and in case of Israel, is the 8. So that is why the Israel technology is very much superior all over the world. It is a small country and surrounded by all the countries are enemy to Israel, but still it is surviving because of its technology. That is, they are very much, Israel technology is adopted by India and other countries. The USA also adopts Israel technology. So a country which is very much well into research, that is a strong country. So Israel, being a small, very small, it is maybe smaller than any of our states, Indian states, but still it is so powerful because of its research. So that research facilities is encouraged under this new education regulation policy, and a budget outlay of 20,000 crores. Government of India is going to earmark for the research, and that will be given to these institutions after the new body comes into picture. So these are the different features of this national education policy. It is a very big document. The document is 500 pages, but these are the things which I have taken as extracts from the which are relevant to our pharmacy education or higher education. So these are the research grants will be around 20,000 crores, and NRF structure will be a good organized structure headed by a chairman. That will be a separate body. And these bodies also, DBT, ISTE, ICMR, UGC also, they will fund for the research funding. They will give after rigorous review to build the research capacity of all the university and colleges of this country. So this is about the salient features of the National Education Policy 2020 and the relevant portion of this higher education and technical education. I have just briefed you. So because more it is a 500-page document, and most of the documents are related to elementary and your school education, and they have modified that, and they have also modified this multiple regulatory authorities they are going to do away with, and they will come with a single regulatory authority for all the technical courses as well as higher education. So this is about National Education Policy 2020, which may be helpful to the faculty members and other related stakeholders for their future planning in research, and the different people who are running different institutions. This information may be helpful to all of them. So this is my total presentation. I thank you, all of you. Thank you. Thank you.
Thank you, sir. Thank you for sharing your views and updates on National Education Policy 2020. It has really enlightened us on the future aspects of changing scenario in the field of education. So, sir, thank you very much, and please stay with us. Over to Subhasri, madam.
Now I request sir to stop sharing the slide presentation.
Stop sharing, huh? Yes. Now I have stopped that sharing, now it has come.
Yes, sir. Yes, sir.
Okay. Okay. So, namaskar, Dr. Patnaik. Very nice to see you today evening.
Thank you. How's your health?
Feeling better. Actually, today morning I took medicine, so feeling better. So very happy that your institution is organizing good seminars, and in this along with the ISTE, this is a good seminar. I found good speakers has come, and all these will add value to your faculty members and students. It's a good initiative by you.
We are planning to have more seminars. At least.
Oh, good.
seminars in a year.
Yes, yes. Because your institute is the leading institute in that part of Odisha, leading and very old institution. So you will show the way to other people that yes, this is the what is called technical education or pharmacy education. So you will be a distinguished leader in that area in case of pharmacy education.
Let us see. We are trying.
Yes, 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 yes.
It goes with the change of the situation, time, position, everything. Corona came, pharmacy has gone up. Something else.
The corona, because of this corona, the identity of this pharmacy education has been established. You see, earlier people were thinking that what is pharmacy? When entire world was looking for a pharmacy organization to come out with one vaccine. So ultimately, which came, you see, Bharat Biotech or Serum Institute of India. What are they? They are pharmaceutical organizations. They came with this vaccine. Not any AIMS did not came with the vaccine. So you see, compare AIMS, that is a different field, and pharmacy is a different field. And this Bharat Biotech, this Serum Institute of India, do you know the owner of Serum Institute of India, Poonawalla? Directly he speaks to Prime Minister, because the Prime Minister is so dependent on this vaccine. So you see the stature he got during this corona period. And Poonawalla, he has spread his business all over the world. India is supplying your vaccines to all the Asian countries, European countries, except Pakistan and China. They are in a queue to purchase vaccines from India, Brazil, Argentina, Chile. They are purchasing vaccine from India only. So see the importance of pharmacy at the end.
There will be a lot of development.
Definitely.
The disease may change, vaccine has to change again.
Yes, but these pharmacy organizations, pharmaceutical manufacturing organizations, they will continue to grow.
Yeah. In other countries, pharmacy is on the top.
Yes, yes.
Lately coming up. Okay, please take care.
Thank you very much, sir. Now it's the time for the distribution of different awards, and to the participants, those who have participated in this international conference on new horizons in pharmaceutical research, ICNSPR 2021. Now I am going to share something, my views. So good afternoon to all the honorable guests present in the award ceremony of ICNHPR 2021. I am Professor Dr. Subhasri Sahoo, the host of this evening. I feel honored to share the participants' achievements and celebrate all of the extraordinary performances of participants. With excellent performances, all of the participants proved that everybody is a winner. I congratulate all of the participants for reaching this final level, and I give my best wishes to them for the result of this award ceremony. The result will be out soon, which I will announce in this ceremony on behalf of ICNHPR 2021. Every participant has some extraordinary quality who will make an appropriate role model in the future pharmaceutical research. Despite their various interests, I am very honored to welcome our guest of the evening, Professor Dr. Pravat Kumar Sahoo, sir, Director, DIPSAR, New Delhi, who has who does not need any formal introduction because everybody is familiar with his achievements, developments, and improvements in the India's pharma research. Sir has the initial inspiration for us for the conducting this international conference. Sir has initiated and advised me to talk with our chairman, sir, KANAK MANJARI Institute of Pharmaceutical Sciences, ROURKELA, Dr. Biswa Patnaik, sir, to discuss about the organizing this international conference. So thank you very much, sir, for your kind advice. By following your words, we can organize such type of program. I want to welcome the committee members of ICNHPR 2021, our honorable chairman, sir, Dr. Biswa Patnaik, sir, our vice chairman, Dr. Sri Patnaik, and all members in this prestigious award ceremony. Regarding these awards, the awarding committee is nothing but the 3 members are there, distinguished members are there. Professor Dr. Pravat Kumar Sahoo, Director DIPSAR, New Delhi, Dr. Biswa Patnaik, Chairman, KANAK MANJARI Institute of Pharmaceutical Sciences, ROURKELA, and Professor Dr. Bhupen Chandra Behra, sir, Professor, Department of Pharmaceutics, KANAK MANJARI Institute of Pharmaceutical Sciences, ROURKELA. Now, on behalf of the ICNHPR 2021, I am going to declare the name of the participants, those who are having their excellent performances. All participants will receive their participation certificate, but some participants, the awarding committee have selected to whom the certificate of excellence should be awarded. Now I am sharing the certificates, and sir will felicitate both Pravat Kumar Sahoo sir, Dr. Pravat Kumar Sahoo sir, and Dr. Biswa Patnaik sir will felicitate the participants. First one is Prasanjit Mohanty for poster presentation, certificate of excellence.
Please accept, accept the certificate of appreciation.
Thank you, madam.
Next participant is Monika Das.
Manas Das. Please accept your certificate of appreciation.
Thank you, sir. Thank you, ma'am. Thank you so much.
Next is Monika Datta. Are you
Thank
here?
you. Yes, ma'am. Thank you, ma'am. Thank you, sir.
Congratulations.
Muti Malika Datta.
Yes, sir.
Accept certificate of appreciation.
Thank you, sir.
Yes.
Thank you, sir.
Anushka Das.
Yes, sir.
Congratulations,
Thank you so much, sir. Thank you, sir.
Anushka Das.
Thank you, sir.
Amitarnath Chatterjee, are you here? Amitarnath Chatterjee?
Yes, ma'am.
Amitabh Chatterjee, your certificate of appreciation. Please accept it.
Thank you, sir.
Congratulations, Chatterjee.
Priyabrata Panda, are you here? Priyabrata Panda?
Yes, ma'am. Yes, ma'am.
Priyabata Panda, please accept your certificate of appreciation.
Thank you, sir.
Ritusmita Rautray, your certificate of excellence. Please accept it. Ritusmita Rautray.
Yes, sir. Thank you, sir.
Certificate of excellence. Accept.
Thank you, sir. Thank you, ma'am.
Miss Binodini Sect X.
Yes, sir.
Binodini
Yes.
Kesh. Binodini Kesh. Certificate of excellence. Please accept. Spelling is XESS.
Thank you, sir.
Huh.
Blessy TM, please accept your certificate of excellence.
Thank you so much, sir.
Niluram Parida, sir.
Thank you.
Please accept your certificate.
Thank you, sir. Namaskar, sir.
Namaskar. Sarojini Naik, please accept the certificate of excellence.
Thank you, sir.
Varsagiri, are you present here?
Yes, ma'am.
Basha, please accept your certificate
you.
of excellence.
Thank you, sir.
Priyanashmi Chaudhary, please accept your certificate of excellence.
Thank you, sir. Certificate.
Anjulata Manjari Sahoo, please accept certificate of excellence. Anjulata Manjari Sahoo.
Madam, are you here?
Mrs. Manjari Tirke, please accept your certificate of excellence. Manjari? Manjari Tirke?
Yes.
Subhanarayan Mahapatra, accept your certificate of excellence.
Thank you so much, sir.
Thank you.
Thank you so much, sir.
Rohit Agarwal, accept your certificate of excellence.
Thank you, sir.
Vikas, accept your best research award, scholar award.
So here I like to tell something regarding the best research scholar award to Vikas Das. He has very much a good young scientist. That means he is performing better. He has some publications also, and now he is the scholar BPUT PhD scholar now. Vikas Das, are you here?
Thank you, sir. Thank you, madam, for the kind guidance. And thank you, sir.
Vikas,
Thank you, sir.
please accept it.
Here I want to interrupt. I want to tell one thing, that why so many excellent certificates are there. As in our conference, the main theme and different sub themes are there. Considering the sub themes, we have decided the organizing committee. That means the award committee has decided these certificates. That means different sections are there, subsections are there. Considering the sections and subsections, we have decided the certificates.
Dr. Manoj Kumar Dalai, please accept your certificate for Distinguished Scientist Award.
Sir, namaskar. It feels privilege to receive this certificate. Thank you, sir.
Thank you. Professor Dr. Bhupen Chandra Behra, please accept your certificate for Distinguished Award. Dr. Behra, certificate is issued for Distinguished Scientist Award.
Thank you, sir.
Professor Dr. Yeah. So this certificate is given to you for Lifetime Achievement Award.
Sir, namaskar. Thank you very much, sir. It's a privilege, honor to get this award from you. And also thank you very much, Dr. Pravat Kumar Sahoo, sir. Sir, namaskar for being a part of this conference. Thank you very much, sir. Thank you.
Thank you. Thank you.
Here I am like to tell something about our principal sir, Professor Dr. Biswa Behera sir, regarding this award committee, why I decided to give this high esteemed award to Professor Dr. Biswa Behera sir, as he is currently working as principal of KANAK Institute of Pharmaceutical Sciences, ROURKELA. He has completed his B.Pharm and M.Pharm and PhD. He has more than 23 years of teaching experience, published more than 15 research papers in national and international journals, and more than 18 papers abstracted in national and international journals. He is a life member of Indian Hospital Pharmacist Association, Association of Pharmaceutical Teachers of India, International Society of Teachers and Researchers in Chemistry, and attended many FDP, refresher course, seminars and workshops. He has guided more than 15 MPharm students, and 3 students are pursued PhD under his guidance. He has written a book chapter, Perspective of Indian Nephroprotective Medicinal Plants in Research Trends in Medicinal Plant Sciences, Volume 9, published by Akiniki Publication, Delhi. And after during this principal by this KANAK Manjari of the KANAK Manjari Institute of Pharmaceutical Sciences, ROURKELA, Dr. Behra sir, during his period, the college has developed a lot. We have got 100 seats in BPharm, and NCR, that means Nodal Center of Research, is approved by BPUT, and also he has applied for NCR for FDP and NCR for industrial collaboration. So many initiative steps are taken by our principal, Professor Dr. Biswa Behra sir. And he is the principal in different colleges. Now he's the principal of KANAK Manjari Institute of Pharmaceutical Sciences, ROURKELA. This college is also developing and on ongoing progress in different aspects. This institute is going on. Thank you very much, sir, for your effort.
Thank you, madam. Thank you for the word of appreciation, madam. Thank you very much.
Now it's time to appreciate our Vice Chairman, KANAK Manjari Institute of Pharmaceutical Sciences, ROURKELA, Dr. Sri Patnaik. Sri Patnaik, madam, is a gold medalist. Now she is the Vice Chairman of KANAK Manjari Institute of Pharmaceutical Sciences, ROURKELA. Madam, having noble personality, when I have requested for any help from her, he has extended her hand for the institute for different programs, having conferences, seminars, and any help when we have extended and we have told and we have requested. And Madam has always cooperative, very nice personality, and she is the patron of this international conference. Now we are going to felicitate Dr. Patnaik, madam.
Thank you, ma'am. Nevertheless, I'm very glad, and it's a huge honor for me to receive this on such an important and momentous occasion of my life. I'm deeply honored. Thank you, sir, and KANAK Manjari Institute of Pharmaceutical Sciences. Wishing KANAK Manjari Institute of Pharmaceutical Sciences, ROURKELA, oceans of good luck and many more international conferences and national conferences to conduct. Thank you so much.
Dr. Prakash Kumar Dalai. Congratulations. So the certificate issued for you for your work.
Thank you, sir.
Dr. Manoj Kumar Mahapatra. So you are awarded. The certificate is issued to you as Young Scientist Award.
Thank you,
Please
sir.
accept.
Thank you very much. Thank you, sir.
Professor Subhasri Sahoo, Dr. Subhasri Sahoo, please accept the certificate for Young Women Scientist Award. Please accept it.
Congratulations, Dr. Subhasri.
Thank you very much, sir. Professor Dr. Pravat Kumar Sahoo, sir, esteemed Honorable Chairman, sir, Dr. Biswa Patnaik, sir, Vice Chairman, Dr. Sri Patnaik, Madam. It's my privilege to receive this esteemed award, Young Women Scientist. I think in my life, if I will try also, I can't receive this type of certificate. And I am very happy to receive this certificate from such a great person, Director DIPSAR, Dr. Pravat Kumar Sahoo, sir. He is my teacher, my inspiration, and Dr. Biswa Patnaik, sir. Always he has inspired me and always encouraging me to do so many things. Thank you very much, sir, for awarding me this type of certificate, and it is matters a lot in my professional career. Thank you very much, sir.
Thank you. Thank you. Congratulations.
Now it's time for to felicitate our guest, Professor Dr. Pravat Kumar Sahoo, sir. So I request our chairman, sir, Dr. Biswa Patnaik, sir, to felicitate to Sahoo, sir.
Dr. Sahoo, we are very much
thank you.
proud of you, and this is a token of our respect. Please accept the
Thank you.
certificate.
Thank you. Thank you. Actually, I wish to congratulate respected B.R. Patnaik, the chairman of the institute, who has taken so much of initiative to organize such a nice conference. I also congratulate all the award winners, certificate winners, and I congratulate all the organizers and participants for attending this conference. And I especially congratulate Dr. Subhasri Sahoo, who is taking a leading role for organizing this type of seminar with ISTE. This type of seminar will bring the name and fame to the institution, and no doubt the institution is already a leading organization in that part of Orissa, and further it will strengthen the institution, and it will motivate all the researchers and all the. And I also congratulate Professor Biswa Behera, the principal of the institute, who has taken keen interest for organizing this type of seminar. And I hope I am really very much thankful to Subhasri. Always she insists and invites, sir, you have to attend the seminar conference. So I always try to encourage her that yes, I will attend. So last time also there was a seminar I attended, and this conference also. Yesterday I was in AICTE for some official work from morning to evening. I could not attend, and today, as per my promise, I could attend this, and very happy. And mostly I find that the chairman of the organization, Dr. B.R. Patnaik, he is also encouraging the entire team to initiate this type of programs, which will help the institution, students, staff, and it will give gross value addition to the institution in building the name and fame and giving quality education and research in the field of pharmaceutical sciences. I thank you, all of you, for inviting me to attend this conference. Thank you, Subhasri.
Thank you, sir. Sir, it's well that we will show the certificate of the participants also. That means it will be a recording for us. Now it's the time for vote of thanks. Good evening, all dignitaries, and for flagging of ICNHPR 2021, I feel honored and privileged to get the opportunity to propose a vote of thanks on this special day. This is the proudest day in the history of our college, KANAK MANJARI Institute of Pharmaceutical Sciences, Szczecin, ROURKELA, India, as we have hosted this biggest international conference in Odisha, India, with many delegates and participating colleges and universities across all streams of education, from science to technology to research. I would like to thank Indian Society of Technical Education India for association with us for organizing this conference. I would like to extend my special thanks to Dr. Biswa Patnaik, Chairman, KANAK MANJARI Institute of Pharmaceutical Sciences, ROURKELA, for his support, relentless effort, and encouragement to organize International Conference on New Horizons on Pharmaceutical Research 2021. On behalf of this KMIPS ROURKELA, I convey my regards and hearty thanks to our chief guest, Professor Dr. Chittaranjan Tripathi, Honorable Vice Chancellor, Biju Patnaik University of Technology, ROURKELA, Odisha, India. I also like to thank all the honorable delegates who blessed us with their presence. The guest of honor, Dr. Namrata Pathak, Director, Scientist F and Associate Head, Technology Mission Division, Nano Mission, Department of Science and Technology, New Delhi. Guest of honor, Sri Himanshu Shekhar Behera, OSS, Registrar, Biju Patnaik University of Technology, Rourkela, Odisha, India. Dr. Biswaranjan Patnaik, Honorable Chairman and Chief Patron, Dr. Sri Patnaik and Mr. Abhishek Patnaik, humble Vice Chairman and Patron. I accord my sincere gratitude to Professor Dr. Pravat Kumar Sahoo, Sir, Director, Delhi Institute of Pharmaceutical Sciences and Research, DIPSAR, University of Delhi, New Delhi, India, ex-Director, AICTE from 2010 to 2013 and 1998 to 2004, ex-Director, Continuing Education Program of AICTE, Professor Dr. Monika Machoy, Division of Orthodontics, Pomeranian Medical University in Szczecin, Poland. Professor Dr. Kunal Pal, Department of Biotechnology and Medical Engineering, National Institute of Technology, Rourkela, Odisha, India. Dr. Suranga Vikramachi, Department of Chemistry, University of Kelania, Sri Lanka. Dr. Indranil Banerjee, Department of Bioscience and Bioengineering, Indian Institute of Technology, Jodhpur, India. Professor Dr. Slomir Wilkinski, Department of Basic Biomedical Science, Faculty of Pharmaceutical Sciences and Medical University of Silesia, Katowice, Poland, for taking time from its busy schedule to grace ICNHPR 2021 with their insightful deliberation as well as sharing valuable ideas regarding new innovations in pharmaceutical sciences. At the outset, I would like to thank Professor Dr. Subhakant Das Sir, Principal, Regional Institute of Pharmaceutical Sciences and Technology, West Tripura, India. Professor Dr. Chandrakanti Chakravarti, School of Pharmacy, the Nyotia University, West Bengal, India. Dr. Manas Ranjan Naik, Professor in Charge, Research and Development, Biju Patnaik University of Technology, Rourkela, India. Professor Dr. S.N. Sinha, Ex-Deputy Director, Indian Jute Industries Research Association, West Bengal, India, for their guidance to organize this ICNHPR 2021. I thank all colleagues and technical teams who have significantly contributed in diverse roles and capabilities to organize ICNHPR 2021. I will be failing in my duty if I don't acknowledge and appreciate all the members of organizing committee without whose dedication, spirit, this ICNHPR could not have its grand success. To our principal sir, Professor Dr. Biswaranjan Behera sir, Professor Department of Pharmaceutics, Professor Dr. Bhupen Chandra Behera sir, Professor Dr. Manoj Kumar Dalai sir, then Dr. P.K. Polai, Dr. Manoj Kumar Mahapatra sir, Dr. Itisri Mahanti, Dr. Sarmishta Naik, and all patrons, that means Vice Chairman Dr. Sri Patnaik and Mr. Abhishek Patnaik and our Chief Patron Dr. Biswaranjan Patnaik. We also, we from KMIPS Rourkela affiliated to BPUT Odisha, India, and participants from other participating universities and colleges got lots of things out of this conference. Words are not enough to tell you how this conference is going to help us in forming our curriculum and pharmaceutical research strategies. I personally like to thank Dr. Namrata Madam's speech, where she mentioned the need for pharmaceutical research. The way she explained the topic is explanatory. I would like to extend my heartfelt thanks to all participants for being with us over the 2 days, adding value to our program. I would like to thank all people who have been instrumental to IC NHPR 2021. I'd like to thank the Siddharth Shankar Bera, then Prasann Kumar Nanda, Ipsita Rai, Sanjukta Das, Priyanka Kanungo, and Abhay Kumar Swain for technical assistance and all support, and all staffs of KANAK MANJARI Institute of Pharmaceutical Sciences, both teaching, non-teaching staffs, along with all staffs starting from the all are supporting us for organizing this this type of program, international conference, and this is the first in our history of our institution. I would like to thank as the organizing secretary of this conference, Professor Dr. Subhasri Sahoo, HOD, Department of Pharmaceutical Sciences, ROURKELA. It will be my, that means I am very thankful to all those who have supported me, and also to this organizing committee for organizing such a nice program. Thank you very much. Now it's time to end our program.
So, namaskar sir, Royal sir, Pravat sir, namaskar. Namaskar.
Namaste.
Sir, sir left. Sir left.